Hello, everyone, and welcome to the Neural Network, a podcast collaboration between Grey Matter's Journal and Connect. I'm Arshia, along with Hutch.
Hello.
And we also have Shrishti, who unfortunately wasn't able to be here today. We'll be your hosts for today's episode. Before we get started, I wanted to tell you guys more about this collaboration. Podcasting is a fantastic way to create conversations around interesting research in frontiers such as engineering, AI, and neuroscience. It allows people to interact with a spectrum of perspectives to develop a greater understanding of these topics.
This collaboration hopes to spur interest in neuroengineering related topics and provide new avenues for those in the community to get involved with work that's fundamentally changing the way we live. The episodes we produce will contain interviews of graduate students, postdocs, PIs, and others involved in neuroengineering research. As we interview these experts, we take care to delve into the nuances of their work and connect it with our own lives. With that being said, we hope you enjoy these conversations and learn about the amazing work that these scientists are pioneering.
Today we welcome Dr. Kevan Kidder, a researcher of the Basso Lab here at the University of Washington. As a PhD student, Dr. Kidder focused on the roles of the medial prefrontal cortex and hippocampus in spatial working memory abilities. While at the Basso Lab, Dr. Kidder works on creating methods that can help discovering the psychological and cognitive markers for neurodegenerative diseases using primate models. Thank you for coming and talking with us.
You're welcome. Glad to be here.
So right off the bat, we were wondering if you could elaborate on how some of the work you've done prior led to where you are now at Basso Labs.
Cool. Excellent. Well, I guess first I'll just start with talking about what I actually do there right now. So I've been with the Basso Lab only 7 months now because I got my PhD back in December of 2022. And in the Basso Lab, it's been just a great place to work. That's really challenged me in a lot of ways because it's different from a lot of what I did in grad school. But now I work with primates every day, and I get to hold their hands while I'm doing cognitive training with others. And some days I'm like, I can't believe this is my job. So it's been pretty amazing. And the work we are doing there revolves around, I have two big projects that we're working on. One is around creating a new monkey model of Parkinson's disease. In fact, if people didn't know, there is it seems that non-human primates, as we call them, NHPs, don't actually get Parkinson's or Alzheimer's disease, even though they have a lot of the markers for it. So in the past, a lot of the tests and therapies that we've developed using Parkinson's based off of non-human primate models, those animals didn't actually have Parkinson's. They were what we call toxin-based, where we would give them neurotoxins, which would actually damage their dopamine neurons. And so you can mimic, by damaging those dopamine neurons in the substantia nigra, you can mimic a lot of the things that we see in Parkinson's, but it's not actually Parkinson's. Don't get me wrong, a lot of really cool therapies came out of those models, such as deep brain stimulation to help treat Parkinson's came out of that. But now in the Basso Lab, we're actually trying to see if we can induce something that's more naturalistic and closer to Parkinson's as humans would have it, which evolves over decades. And then the other project we're working on actually looks at just the natural aging process in general, where my role as a researcher there and engineer is actually to engineer these cage-side systems, which are these rigs that allow us to do cognitive testing on the animals. So basically we hook these big devices with touchscreen monitors onto the animal's home cage. And the animals, they're not restrained in any way. They are totally allowed free access for hours to play this video game basically as much as they want. And they actually love it. They actually really like it because they get little juice squirts, apple juice or grape juice or cranberry juice every time they get it right. And they really like playing the little touchscreen games, which test different aspects of their cognition. So my engineering role there comes into play because then you have to engineer and work with different physics shops here at UW and get plexiglass made and figure out the right ways to put electronics together that makes it safe for the animal but also gets what we want. And then my scientist side comes in, and then that's when I analyze the data. There's a lot of data in all of science now. I'm sure you guys are aware. So we're all data scientists, it seems like nowadays. And then we also collect fluid samples, just like you would go to the doctor to have blood tests run. We do the same thing to the animals, and we're trying to analyze, or we're working on pipelines to analyze immunological markers, so like T cells and B cells and monocytes, along with looking at gene expression through mRNA-seq, so we can see what genes are active and not active, And then we are also looking at cytokines. So what is their level of inflammation? And we just took a new master's student to throw in some microbiome research by collecting fecal samples. So as you can see, there's like 4 big data sets there on top of the cognitive data sets. And we're hoping we can, at the end of the study, correlate their cognition between young and old non-human primates, look at all of these biomarkers, and specifically look for genes and biomarkers for diseases like Alzheimer's or dementia. And hopefully the idea would be to be able to identify biomarkers that would help us predict either what we call MCI, mild cognitive decline or impairment, or Alzheimer's and dementia 10 years before any clinically relevant symptoms would occur. So my engineer part, I get to tinker with a lot of, just a lot of hands-on shop stuff and electronics. And then the science part comes to making sure all of this is done in the proper way so that it tests at what we actually want to in the cognitive domains.
Sounds like a sweet deal for everyone involved.
Yep.
Wow.
Yep.
Yeah. So that's about Basolab, which is really interesting.
Mm-hmm.
But so what are some of your other interests or like any other projects that you've been doing in the past? Or
In the past, yeah. So this is where, very recently, like I said, about a year ago, I just got out of the Mizumori lab, which is the Laboratory of Neural Systems, Decision Science, Learning, and Memory. And that lab was a lot more, and it's close to my heart. Shout out Sherry Mizumori. She was my mentor for about 8 years through undergrad and grad school. And we did a lot more basic neuroscience. So I got to, I became an expert on the medial prefrontal cortex and hippocampus. And I got to do a lot of different projects where I was basically trying to look at underlying neural mechanisms that would allow for information transfer between these two areas. Because as I grew to learn in grad school, any decision you make in life requires some sort of information. Because if you're not making an informed decision, like what are you making the decision based off, right? You're basically having a reflex. So any decision that you make, whether what type of food you want to go eat today for lunch, what route you're going to take to work, is based off of something you're seeing and some sort of information, and that information is memory, really. So the hippocampus may be important for recalling a memory, but it needs to know what to do. What use is that memory by itself? And that's where you have the medial prefrontal cortex now trying to it knows the strategy or what wants to be accomplished, and so it needs to work with that memory center, the hippocampus, in order to get the right memories recalled and then utilize that memory in the right way. So a lot of that so I did multiple projects looking specifically and picking apart when are these two brain areas actually communicating with each other, and trying to create cool causal experiments that aren't just correlational. That was the cool thing about that basic neuroscience we did in that lab. I really was trying to get more at causation by doing experimental manipulations more than just like correlating a few data points together.
Awesome. I don't mean to backtrack on that, but
yeah, please. Yeah, I know I said a lot.
No, yeah. It's just part of what you just said that stuck out to me was this very extended relationship with a mentor that you've had from undergrad all the way up until grad school, which seems, if you could maybe elaborate on some of that, maybe how that relationship helped drive you towards where you are right now. Anything?
Absolutely. So let's take it back to early Kevan when I was figuring stuff out. If that's okay, I'll digress on a little bit of how I got here. Now I'm happy to say, like, I feel like I have a lot of accolades, and I've made it most of the way to where I want to go. But in 2008, I graduated high school, and then I applied to UW and Wazoo, and didn't get into UW, went to Wazoo, and I actually flunked out of Wazoo 3/4 of the way through with a 0.6 GPA.
Oh.
Yep, not the greatest time in my life. Then I moved home with my parents. Thankfully, I had very supportive parents. And everyone knew I was supposed to do something science-oriented. I knew I wanted to be a science, but my mind just wasn't there. I actually then tried immediately moving home with my parents to go back to community college, or to go to community college, and I flunked out of that again. I was not ready. And so I finally figured, okay, you're not ready. And I took 2 or 3 years off and soul-searched. And my mom bought me a subscription to Scientific American Psychology, I think, and another one, Scientific American. I tell you, I read those every single month for those 3 years. So by the time I, a few years later, went to community college to get my AA, I knew I wanted to study the brain. Like, that was set. That was not a question. I didn't know where I wanted to go, but I knew I want to study the brain because it's what creates your reality. Like, I used to want to do astrophysics and all the crazy things out there, like dark matter. And then I'd be looking up and I'd be like, but in here is what's making my entire perception of any of that. And if we don't understand that, why am I going to focus on this stuff out there? So I had that realization, kind of epiphany, during my soul-searching years. So after I finished my AA at Pierce Community College, the AA in psychology, when I got into UW, I was finally accepted to UW. So I started as like a sophomore, I guess, credit-wise. And I knew I wanted to do research, but I didn't know what exactly. And actually, I joined a research lab in Dr. Stephen Buck's Laboratory of Human Color Vision right away. I got a little bit of the ground, how it works for research, but I actually, after that first quarter, I joined the psychology honors program here at UW, and that is where I met my first mentor, Philip Baker, who was the postdoc in the Mizumori lab. And he was talking about optogenetics. And optogenetics, for anyone who doesn't know, is a way that we can control neurons with light on millisecond timescales. And to me, this was almost 10 years ago. I thought it was the coolest freaking thing that I've ever heard of. And I was like,
still is.
I was like, I need to do that. I was like, please let me join my lab. I got a tour of the lab, and Phil, the postdoc in the lab, was actually who I consider my first mentor. And he was, I didn't have much contact with Sherry at that time because she was actually the chair of the psychology department, which anyone who's been in that situation, they're really busy. But so Phil was my mentor for a few years, and then eventually I ended up getting into grad school. It took me 2 years in a row of applying to grad school to finally find a home somewhere. But Sherry took me under her wing, and that's when I started interacting with Sherry Mizumori a lot more in grad school. And she really took me under her wing, and I have great relationships with both of these people still today. Think about them every day. And I want to say, for anyone who's out there thinking about going this path, having a mentor is the most valuable thing that you could possibly do at all. Because for me, there was no doctors in my family. The only doctor I ever knew were the doctors I went to when I had a cough. So this was totally new grounds, and I learned along the way that it really is like a networking game you have to play, and the ways to get into grad school. And I learned all of the techniques. I learned how to effectively write as a scientist through my mentors and their great teachings. So mentorship is huge. Find yourself a mentor, definitely.
That's an incredibly inspiring story.
Yeah, thank you.
Yeah, no, good on you, dude.
Thank you.
Obviously, you have a great amount of passion for neuroscience as a whole, but we were both curious as to what other parts of research sort of keep you engaged. Like, why do research as opposed to someone with your skill set could reasonably go into industry or kind of do anything else? So what is it about research that sort of has a grip on you?
No, that's great. So I really like, some people will talk about scientists have a little bit of ego. And at this point, I have 5 or 6 scientific publications, 2 first authors, and I love having my name on those publications, don't get me wrong. And I think that's so cool. And overall, I have felt at home in academia, and that's all I've known. And so far where I'm at in my life, the questions you're asking me are something I kind of ask myself every day. Because by going and being a research scientist right away, I made that decision for a couple reasons right now. One, it makes more money than a postdoc. And I have, you go into some debt sometimes. It's hard. When you're a grad student, you are paid minimum wage. And so, I got tired of living that way, and I just wanted I wanted to reap some of the benefits, of working this hard for 10 years. So that's one reason why I went straight to research scientist. It used to always be my dream to be a professor. That's something I still ponder about every day, but I see professors really busy, really freaking busy. And so my job, I go home, I don't have to think about work, and I'm loving that because it was not that way in grad school, as you can imagine. Versus the industry, industry jobs will pay equivalently to what I'm being paid now as a research scientist. So I may go to the industry. I think about things like going and working for Elon Musk at Neuralink, because he was asking a few years ago. I watched like a 2-hour special on him and all the different types of scientists, and he looked at their ePhys electrophysiology acquisition system they were using, and it was OpenEPhys, or at least a version of it, which is exactly what we used. It's an open source ePhys acquisition system. I used that through grad school, and I was like, oh, it'd be so cool to work there. So I think about the industry a lot still, and at this point in my life, I think I'm gonna sit here in this lab and get some cool things done for a few years, and then relook at what's out there and options. Yeah, but the thing with industry, right, is that you don't actually own any of your research. You're more, you're one of the worker bees to this greater picture, which means their pipelines are really efficient. But you only do certain things. For instance, going and working at the Allen Institute, who one of my star undergrads, Ryan Gillis, he currently works at the Allen. And from my understanding, his job is just to, there's these things called neural pixels. Are you guys aware of neural pixels?
No,
No, those are high density electrode arrays, state of the art right now, where you have 1,000 different channels, hundreds to 1,000 of different little electrode channels. So it allows us to collect so much data from animals on a level that hasn't been seen before. So they're like these small little probes. They look tiny, but there's like thousands of channels along the surface of them. So you get lots of data. And so their team, from my understanding, right, their one job is to prep an animal for surgery, put that little thing in, and remove it. And there's so much planning in that one little piece. Whereas now, in my type of job, I have more of my hands kind of in every aspect of the project, and I like that more.
Yeah.
So that's one thing I like about this type of stuff.
Yeah. Do you think that Allen Institute work you were talking about has something to do with the current connectomics areas at all?
The research I was specifically, like my star undergrad,
or what you were talking about with like the neuropixels, because I recently attended a talk with someone who was on that sort of team, and he was touting like how valuable this one particular like n=1 data set would be. It was really cool, actually. It's like it's like using machine learning in order to like find the boundaries of like membranes. It's like finding where mitochondria are in the cell, and going through and taking brain slices, incredibly thin ones, like 4 microns or something, incredibly thin brain slices, and kind of connecting or finding all of those neural connections within one animal.
Wow. And
that was really cool.
Yeah, that's crazy. And so I don't quote me on this, but right, because I don't know actually what goes into all of their stuff, but my idea is that whatever goes into that connectome and their brain atlas, they are using pieces from every team that they have to help build that. Whether or not that relates specifically to the connectome, I'm not sure. But for example, I know a big thing in the atlas is just to identify. Now we know there's not just excitatory neurons and inhibitory neurons, right? We know there's all these ranges of different neurons. Those GABAergic interneurons, as we call them, there are 20 to 50, maybe even 100 subtypes of them. And I know, like the electrophysiology side, I know what they're doing and putting in the brain atlas is characterizing what are the firing characteristics of each one of those, and how good can you know that this is a particular subtype of neuron based on just its firing characteristics, right? And the answer is usually kinda. So then you need to bring in molecular signatures, and genes, and looking at where they connected and stuff. So I think it all works together eventually. Yeah,
it's exciting.
Yeah, it is exciting. But the more you get into the brain, the more you're like, holy moly, how are we ever going to figure it out? That's how I felt in grad school. It was like every day I'd learn something new.
Right? I think we keep hearing that there's so much about the brain that we just don't know.
Yeah.
Yeah, but as someone who wants to go and research myself, I have to ask, what are some of the exciting possibilities in neuroscience, or some of the research that's happening that's really interesting for you?
Yeah. Well, I'm just gonna say the first thing that came on my head was, so one of my loves in neuroscience that drove me down this path, which I don't actively study, is consciousness. That is actually a thing that bridged my gap between physics and psychology. There's quantum. I don't know if I should go that way, but there's quantum mechanics. There's this idea with Schrodinger's equation of the conscious observer collapsing the wave function, and that's how we decide whether a photon is a particle or a wave, because the conscious observer collapses the wave function depending how to look at it. And that's how I kind of got started to get into the brain. I was like, whoa, what is consciousness? And so I followed consciousness for a long time, read a lot about it, and actually became kind of the star guest speaker in multiple classes at UW in the psych departments, whether it was biopsychology or Sherry Mizumori's neuroscience of the mind. Multiple years in a row, I was the person giving lectures on the neurobiology of consciousness. And some of the research that's out today on it really excites me, and it really talks about this new paradigm shift where we're at in neuroscience. And it's that the brain is comprised of many different systems. There might be 5 big macro scale brain areas that are all active together. But then 200 milliseconds later, a different network pops on. Like, you may have heard of things like the default mode network. Right? And there's visuospatial networks and attentional networks. But there's this idea that if you were to look over, say, a half an hour and print out a sheet of data that shows what networks are active, you have this thing called a temporal circuit. And we can analyze whether or not a person is conscious versus someone unconscious or with unresponsive wakefulness syndrome based on what areas are turning on and off on a millisecond to a second time scale. And to me, this idea of analyzing systems of brain areas that interact together and switch on and off, to me, that's a really cool place that neuroscience has to go, and they're starting to get to with a lot of fMRI studies. So I think that's really cool, because you can't just study the MPFC by itself anymore. You have to look at how is it working in concert with other areas. And that's different from how neuroscience has been done 50 or 100 years ago. So that's one. Yeah. But I guess the other places that I'm really excited about, consciousness was just my big thing. So just being able to understand what is that thing, I think will be a great thing. But other advances are similar in that we are getting to places where we might have one brain area that we've always looked at, but we realize there's actually 6 different sub areas within that brain area, and then understanding what are the neuronal subtypes within that area, I think. And understanding that is a place where a lot of neuroscience is at currently, but I feel like that is what we really need for a lot of drugs and therapies to work, because we may have been dumping a bunch of serotonin into a brain area to try to help depression, but we didn't know that there was 4 different subtypes of neurons all acting differently. And now if we can start to figure out how they are responding, we can get to an even more accurate level of treatment.
Yeah, for sure.
And then if I have time for one more, the other big one is psychoplasticogens, as we call them in neuroscience, which are more commonly referred to as psychedelics or hallucinogens. Those, I think, this has just such a potential to be groundbreaking to help so many mental illnesses if done in the right settings with the right type of therapy. We call them psychoplasticogens for their ability to alter neural plasticity. And this is what many researchers across the country now are studying, and they're creating entire neuroplasticity institutes to study psychedelics. Because if we can figure out how they are unlocking neuroplasticity and allowing us to more easily rewire someone's brain, and if we can figure out how to control that wiring, we can do great things to ourselves, and like control our lives better.
Yeah, for sure.
So I'm very excited. I was very excited that my last project in grad school, we have a student, Victoria Ivanova-Holmes, who was working on some psilocybin research in order to see if psilocybin could help animals going through morphine withdrawal and morphine addiction. And I had this cool, basically a brain reading device, an electrophysiological rig that I made that recorded from 3 or 4 different brain areas deep within the rodent brain. And we gave him psilocybin while I was recording from their brain, and they were running around on this little decision-making task. And I have the data, and I'm so proud to be a part of that, and probably have some of the first rodent recordings of a rat on psilocybin, which is the active ingredient in magic mushrooms, for those who don't know. So that type of research, I'm so excited to see where it goes.
Yeah.
Yeah. I have a friend who's interested in a lot of that stuff, and like coupling with like therapies. It does seem really promising. And from what I understand with that sort of climate around drug research as a whole, which we don't have to get into because scheduling is really fun, as I'm sure you know.
Well, can I make a comment?
Of course.
So I've done, to me, substances such as psychedelics, to me, the fact that they have such profound impacts on the mind, to me, means there's something profound happening in the mind. And as neuroscientists, why shouldn't we want to understand the neurobiological underpinnings of what happens, right? That's what these drugs and SSRIs were trying and attempting to do. So I had quite a few projects. We were giving rodents cocaine in order to study how cocaine withdrawal affected decision-making, because addicts, when they go through withdrawal, their decision-making, they actually go through a state of heightened decision-making. They're really good at figuring out how to get their fix, right? But that comes at a cost to many other things in their life. So we had a project where we were just seeing how rodents' decision-making abilities changed. And another one was I spent my first year or two of grad school applying for grants to do THC research. And it was hilarious to me that we had a Schedule II license, and we could have cocaine in our lab, but we could not have THC.
Yep.
So we had to use a synthetic agonist that is nothing like the real thing. And it was so powerful that the hippocampus, the electrophysiology, just it turned deep dark blue on the spectrogram, which normally should be full of colors. And I was like, that's when it dawned on me how powerful the cannabinoid system is.
Oh my god.
Too, because it just shut down the memory center. I was like, wow, there's no way. There's no wonder why there's this kind of what's the word I'm looking for? General thought around stoners not having good memory. And I was like, look at the rat's hippocampus. It's not on. You're not encoding any memory. And I saw that firsthand in the lab. So that was cool. But yeah, the whole scheduling thing, I was like, this is ridiculous. This is absolutely nuts that I can't have just THC to study.
Yeah.
But yeah.
We have been a little bit heady for a while in terms of like neuroscience. Ultimately, this is the question I've been most excited for. Cool. We've been talking about all these different subtypes of neurons and how they interact with each other, but I'm curious about you. Okay. Kev.
Yeah.
Dr. Kidder.
Great.
What type of neuron would you be?
Great. I put some thought into this one.
Good.
I would be a GABAergic interneuron or inhibitory neuron.
Interesting. Why?
Great. Because so many people don't understand the importance of these GABAergic interneurons. So a few facts about, I use the term interneuron because that's traditionally what's used, but I should just call it an inhibitory neuron. So your excitatory neurons, your glutamatergic neurons, they're the ones that actually, your memory engram for a certain memory that you have in life. The excitatory neurons are the ones that, they come online and they actually seem to have that code. But without those interneurons, their firing will be sporadic and that information will never get achieved. So you need interneurons to control spike timing of excitatory neurons. Without inhibition, the excitatory neurons would just all be firing all the same time. If every neuron in your brain's firing at the same time, you're having a seizure. It's not good. You need precise control. And another fact around inhibitory neurons is, at least in the frontal cortex, this is one of the reasons why they say people's frontal cortices aren't fully developed until they're in their 20s, is the number of interneuron synaptic connections seems to peak around your mid-20s. Which I think then you can think about your frontal cortex throughout the day, your brain is thinking a lot of things, but you don't act on every thought you have, which is actually a good thing. But as kids, they act on every impulse. So to me, the thing that sort of makes us an adult and a wise thinker, someone who's tactful, someone who doesn't just blurt out random things, or someone who doesn't just get aggressive randomly, is the fact that you have interneurons telling you to halt that. So to me, that's why GABAergic neurons inhibition, this balance between inhibition and excitation, is so important. Yep.
Yeah.
Yep.
Okay, wow. Yeah. So we've spoken about so much research that's happening right now, but yeah, so we can say that it's a fast-paced research field, right?
Absolutely.
So what is it like being in it? Like being in this fast-paced research
In the field. Well, so I've kind of feel like I've been in two fields, because when I was in the Musumori lab, it felt really cool, like I was part of a family, and that we'd go to this cool conference called Park City Winter Conference on Learning and Memory, and you're there with a couple dozen researchers, and you are all that collective group of people. Like, we're the people who study the hippocampus and the MPFC and rodents, and like we're the experts on this particular thing. It was really cool to be a part of that, and to be part of a handful of four other researchers across the country who are just studying this one particular circuit, and we'd come together, and I've loved it. And now I haven't got a chance to go to any conferences for this type of research, but I will say this type of research that I'm in now with the Alzheimer's and aging, there's so many people on it. It's a little daunting. And I do feel the pressure. Like, literally on my Facebook, I see a new science alert article, and I'm like saving it, and then I'm sending it off to people who do our immunological testing. Like, are we able to do this and this? Because the fear is I'm launching on a 5-year project to go through like a cohort of like 40 animals so that we can actually get some high throughput testing and some really good data sets. In the fear is 2 years from now, are they going to discover some other protein that's on some immune cell that is the next best predictor for Alzheimer's, and will we have missed that? So it definitely is a little daunting on that, but since I like to read a lot of scientific journalism already, it's kind of fun, because literally just throughout my day I'll see something and I can just throw it off over there. So that's a lot of fun. Yeah.
I actually had a very similar experience where in like an upper level physiology class I'm taking, we're working on like, treatments for like a neurological disorder, like in our case Parkinson's. And literally not 2 days ago, our teacher came up and was like, hey, people found a way to use like deep brain stimulation to administer like drug-filled little oxygen bubbles directly through the blood-brain barrier.
Wow.
Just by stimulating it, like
Yeah.
opening it up. It's like, whoa, wow, this is...
Did it use ultrasound? Do you know?
I believe.
Oh. That's a new technology I hear a lot about.
Yeah.
Getting drugs through the impermeable blood-brain barrier, BBB. Yeah, that's hard. That's
not so impermeable anymore.
Not so. No.
Yeah. No, it's exciting.
It is exciting.
Yeah.
Mm-hmm.
I guess on that sort of note, when we sent like our list of questions over to you, specifically regarding some of your older work, you mentioned how a lot of the stuff or a lot of the methods that you used in that lab became out of date, like around the time of publication. So how is it keeping up with that? Not obviously, we've mentioned on a knowledgeable standpoint, but in terms of a practicing standpoint, how is that? How do you deal with
that? Absolutely. So it kind of that answer depends on what you're going for, what your science is. So I feel my actual grad work that went into my thesis was based on optogenetics. I did these transient manipulations where I only disrupted the medial prefrontal cortex for a second here and a second over here and a second over there. And I felt that study was so well designed, that paradigm, that not a lot of people were stepping on my toes there. So those methods aren't obsolete, and actually people should still carry on, because optogenetics allows us to do these very discrete manipulations where in the past they lesioned an area with electrolytic shocks. You destroy it. Now you can't tell what the sub-second activity was used for, right? But the paper you're talking about was this. I was just a co-author on this paper. Actually, the first author was Jesse Miles, and he used these machine learning approaches to do some really cool things. One, we had videos of our animals while they were performing on tasks, and we were trying to get the machine learning. Well, we got the machine learning program to just look at the videos itself and say, is the animal turning left or right, and are they doing this this behavior that we call, it's a vacillating behavior, meaning the animal vacillates its head one direction and then goes the other. It's kind of like oh, nope, not that direction. I want to go the other direction. It's called a VTE, and there's a lot of interesting neural correlates of deliberation around that behavior. So we're trying to get machine learning to identify that. And then he also, later in that paper, we were using the machine learning algorithm to detect VTEs, but also look at hippocampal neural oscillations from our EFIS recordings and see, could we predict the animal's behavior just based off of those neural recordings alone? And it worked to an extent. A lot of machine learning classifiers, if you can get up to 90%, you're really good accuracy. This was above chance level, so it worked. But machine learning, as you guys know, and AI evolves so fast that, by the time that paper was published, I even asked Jesse what he thought of it. And he's the one that told me flat out. He's like, those methods are obsolete. And I think as he learned through his grad career more better methods, he would use something different now. So it depends on the area. If you're anything in machine learning, that is an area I think you just got to know that's going to evolve faster. Yeah.
Yeah, for sure. Yeah. So now that we're talking about AI, how do you think the climate surrounding neuroscience research is going to change with AI?
It's a great question. So at my level, I'm not sure how much difference we're going to see in the next 5-10 years because we've already been using this stuff. We've had our own grad students specializing in machine learning to do kind of the larger scale tasks that deal with a lot of larger data sets. Like, we've had this problem for decades already, so you best believe scientists were already on top of how do we deal with all this data. So now I think the places where it may help a lot is kind of more towards the human clinical work. That is where it could, like, talk about what if are you guys familiar with Neuralink, Elon Musk?
Yeah.
Right? We're talking about chips inside people's heads and stuff. Imagine putting AI in that.
I know,
right?
yeah.
So there's this device that, there's a paper that I taught some of my students, and they had this device called a MIMO device. It's MIMO. I don't remember what it stands for, but the idea was this device was placed in your brain, and it recorded the specific neurons that were active during a memory in the hippocampus. And it recorded them and put them on the device, and it could then later stimulate those exact same neurons during a test so that you could help people remember what they had learned a few weeks ago or something. And it actually worked.
Wow.
And this was like 5 or 10 years ago before AI. Can you imagine if you can now have AI figure out all of the little nuances and complexities to that thing?
Yeah.
Right? It's almost kind of scary. But then we get into Black Mirror. What if the AI decides it wants you to remember something else that you didn't plan on it making you remember? But I think
tricks
AI
with the brain then.
Right? Yeah.
That's insane. Yeah.
We're getting a little tight on time, so I'm gonna ask one final question.
Awesome.
Obviously, you're big on consciousness, and given what we've talked about, with these potential for like neural implants to be able to fire some of these circuits.
Yeah.
Where do you think consciousness ends in that relationship?
I think it's just a new story. It's a new chapter. It's a new beginning, right? The merging of it. The idea behind, I'll talk about it again, Neuralink with Elon Musk, is that, it's a scary idea to have the singularity. We're talking back to the Terminator and the Matrix, all these movies where machines overcome humans because they're smarter, right? And Elon Musk will if you watch interviews, he'll talk about he started Neuralink because he was worried about that. That's what's called the singularity, when robots have their own general AI consciousness. And the idea is, I think it's a cool, exciting story to tell that if we can merge with AI and become more, but still retain who we are as humans, that would be really cool. So I hope the story is beginning for us to extend our consciousness to another realm, but still remain who we are as humans.
Yeah.
That's my hope.
Yeah, for sure. Yeah.
Yeah. Well, with that, I think we call it a wrap.
Awesome.
Thank
you
you.
guys.
Thank you
no.
so much.
Yeah.
It was lovely to speak to you.
Yeah, no.
Awesome.
Thank you for all these insights.
Good.
Definitely
Glad to
appreciate your presence here.
help. Awesome.
But yeah, sweet. Okay. Yep, wave bye to the camera. I guess I'll get in.
There
Oh,
we
yeah.
go.
I'll try. Haven't been in frame
You
at all.
You can clip it.
Yeah. No, yeah, of course. Thank you so much for your time. Again.
You're welcome. It's
Do
great
you-
to be here.
Yeah, no.
I can ramble all day about this stuff, honestly.
Listen. Come back for another one.
Oh,
Hey,
yeah.
I'll be here. That'll be great.
We still got so much more to talk about.
We do.
I
We do.
know.
But yeah, no. Okay, I'm gonna call it there.